Hello, dear Botanicas learners. How are you, everybody? So, welcome back to our botany class. So, in this class, in this video module, we are going to discuss two topics, small topics. One is regarding translation, another one is regarding RAPD markers. So, first we will discuss regarding translation. So, first of all, we have to come to know these translation and RAPD markers, these both terms, the names belongs to what type of field? Translation and RAPD markers, the terms belongs to molecular biology. What is molecular biology in the sense? The study of structure and the sequence and the arrangement or the codons and the functions or the features, whatever it may be, in detail of DNA, deoxyribonucleic acid, and also RNA, ribonucleic acid. So these both comes under nucleic acids. So that is known as molecular biology. So here biomolecules are two types: macrobiomolecules, microbiomolecules. Macrobiomolecules in the sense proteins, carbohydrates, lipids, etc. Microbiomolecules in the sense deoxyribonucleic acid and ribonucleic acid, etc. So these two comes under nucleic acids. So regarding these nucleic acids, if we study in depth, in detail, that is comes under molecular biology. Okay, so here the term translation, I think so you are very familiar. Do you know the term replication? Especially DNA replication, semi-conservative method, one of the famous method in the world proposed by the scientist. And do you know transcription? And third one is translation. So here translation in the sense mRNA codes different types of codons. With the help of different types of codons, finally a protein is synthesized, that is known as translation. So mRNA codes synthesis of a type of protein, that mechanism is known as translation. So we knew already in zoology, I hope you learnt already types of proteins and structure of protein, classification and category, etc, etc, etc. Linear chain proteins monomeric, apart from linear chain called multimeric or polymeric. Linear chain proteins, they have only peptide bond in between the amino acids. Proteins are the combination of amino acids. Building blocks or basic structures are amino acids for the proteins. And multi and poly, linear in the sense ionic bond is present, disulfide bond is present, hydrogen bond is present along with peptide bond. So those are complex structure, helical structure or pleated structure, whatever it may be. So different types of proteins, that is a further topic and another topic. So here our translation concept is mRNA code the synthesis of protein, that is known as translation. So here RNA, ribonucleic acid, again different subtypes. mRNA, messenger RNA, M in the sense messenger, and one more is there, tRNA, transfer RNA. T in the sense transfer, M in the sense messenger, and one more is there, rRNA, ribosomal RNA. Do you have an idea of ribosome? Ribosomes are present in both plant and animal cell. Those are known as protein factories because they will synthesize, produce the proteins. Those ribosomes are produced by nucleolus. So nucleolus is known as factory of protein factories. So here ribosomes are also known as microcell organelles, very minute than the all type of cell organelles present in the plant and animal cell. So here ribosomes, with the help of ribosomes, and the RNA is called ribosomal RNA. Finally, proteins are synthesized. So in that purpose, in that process, tRNA and mRNA are highly helpful. Let's see the mechanism briefly. So once again, what is translation? Messenger RNA code the different types of codons or codings for the synthesis of protein is known as translation. So here in a cell of living organism, especially higher organisms, especially human body cells, they have DNA, deoxyribonucleic acid. This DNA, it synthesize or form the messenger RNA. So in this messenger RNA, the DNA, what type of protein should be synthesized further in the form of message or the codings, it will be implemented. So because that RNA is known as messenger RNA. This messenger RNA later, with the help of ribosomes, it synthesizes a chain of, a linear sequence of amino acids. So these chain of or linear sequence of amino acids together called proteins. Are you clear? So DNA main function is not transferring the characteristics, not transferring the features from the parents to young ones or generations to generation. That is secondary. The primary function is characteristic feature of the DNA is synthesizing the proteins which are perfect or acceptable by the particular cell in the body, then only the cell can survive, then they will be functioning different types of metabolic activities. So DNA main function is synthesizing the proteins. Are you clear, dear Botanicas learners? So DNA, it send a message what type of protein should be formed in the cell in the form of mRNA. Then mRNA, with the help of ribosomes, finally it forms the chain of amino acids. The chain of amino acids combination make the proteins. So here ribosomes, briefly, ribosomes are the micro cell organelles. They have two subunits. So here 70s ribosomes we have and 80s ribosomes we have. That is an entirely separate topic. Just briefly I am explaining you. So one is a smaller subunit, another one is a larger subunit. So into the smaller subunit, mRNA enters. Into the larger subunit, two molecules of tRNA enters. So here tRNA, transfer RNA main function is providing the ATP, adenosine triphosphate energy for this entire mechanism. Nothing but translation. So this is active reaction, not passive. So here must metabolic energy should be required. So adenosine triphosphate is provided by the tRNA and the entire mechanism will be performed. So into the large subunit, two molecules of tRNA enter. Into the small subunit, one molecule of mRNA enter, and with the help of providing the ATP energy, peptide bond is created. 2 plus 1, one peptide bond. Again the reaction repeated. 2 plus 1, one more peptide bond. 2 plus 1, one more again. 2 plus 1, one more again. Again and again, again and again. Finally, these peptide bonds together form a chain of amino acids. That large chain of amino acid finally known as protein. So this is the mechanism of translation. So here main role played by mRNA. Why? Because from the DNA it should get the message or the codings what type of protein should be formed. Then second one, tRNA, because it should provide the ATP energy for this entire process. Then final one, ribosomes. Without ribosomes, they never will be functioned. So this is, dear Botanicas learners, translation. The next one, RAPD markers. So RAPD markers basically mainly used in the PCR amplification technique, polymerase chain reaction. Do you have an idea of ELISA test, which is performed to detect the human immunodeficiency virus, that AIDS? In AIDS patients, to detect the HIV virus, ELISA test is performed. That ELISA test is purely based on the polymerase chain reaction, PCR. So here amplification nothing but detection. So these RAPD markers are beneficial and used in the PCR amplification to detect the primer. So what is a primer? Primer in the sense a short or a tiny or a small segment of DNA or RNA, that is called primer. Very short. So we knew DNA has a double helix structure. It is a lengthy and long strand. So in that long strand, different types of codings and bondings are already synthesized and fixed and located. So need not to study and detect the entire strand. Just if we can able to detect or find something in the primer, that short DNA or RNA strand, then remaining process will be easier. So what type of process will be easier in the sense, in these forensic departments, investigation department, CID or something, CBI, Central Bureau of Investigation, etc. So in this crime investigation report or else these genomic related crimes or relationships problems, to solve those all, these RAPD and PCR amplification techniques are highly useful and beneficial. How in the sense? So here, with the help of this DNA polymerase, the DNA sample which is collected or which is found somewhere by the experts, so with the help of DNA polymerase, it undergone for the replication. So after replication of the short DNA or the primer with the help of DNA polymerase, finally it will be replicated into large DNA molecule, that is known as amplification. So we got short and very less sample or nothing but primer. With the help of polymerase, during the replication or due to the replication, finally large DNA molecule we are getting, that is known as amplification. Finally, after getting that double helix or double stranded DNA, if we expose that to ethidium bromide and UV light, whichever, whatever the expert they would like to find out or detect, that result we will get. So finally, with the help of radiography photo technique, the report, the result will be submitted to the superiors or higher officials, then court will give the judgment. So that is a basic principle in which these RAPD markers are highly beneficial and used and utilized. This entire technique categorized under three main steps or sub-steps. Number one, denaturation. This is not denaturation, denaturation. Second one is annealing. Third one is synthesis. Then after this third step, finally, we will get the result, dear Botanicas learners. So first one, let's see, denaturation. In this denaturation, the DNA segment or the DNA molecule, that strand, it will be allowed for the treatment at the temperature 90 degrees to 94 degrees Celsius. So that is the first step. Remember the temperature range. After that, the second step is annealing. In this second step, this temperature is highly reduced. The DNA strand is cooled down. Whenever this DNA strands from the 94 degrees Celsius to cool down up to 35 degrees Celsius, so the primers present in the strands are joined together. So to give the chance for the primers joined together in the strands, we must cool down. So that step is annealing. Then finally synthesis. Again after cooling down in the third step up to 72 degrees Celsius, we have to provide the warm temperature to the strand. Then finally the complete strand will be synthesized. Likewise new strands, how many we can synthesize in the sense of millions, not hundreds and thousands, dear botanicals learners. Millions of strands can be synthesized. Finally those strands are allowed for ethidium bromide and UV light. Finally the detectable sequence or the bonding is detected by the experts or superiors. So this is the information regarding RAPD markers used in the PCR technique, polymerase chain reaction, and the previous one is a translation. So here in the biomolecule, remember this statement, sentence, dear every botanicas learners. The statement is cell, living cell, inside the living cell DNA, from the DNA RNA formation, that is called transcription. From the RNA protein is formed, that is called translation. Then again from the protein is utilized by the cell. Again from the cell, the DNA again it forms mRNA, from mRNA to protein, again protein to cell. This cycle will be continued. This is known as central dogma. Dear students, this is known as a central dogma. So the entire biomolecules field, that branch of biological science based upon this central dogma. So dear everybody, in one more video we will meet with one more beautiful botanicals topic. Till then, good luck.